Cytotoxic T cells, or CTLs, are a specialized subset of T lymphocytes that act on viral pathogens. CTLs, however, do not act alone. The immunological action against the virus by a CTL is aided by T helper 1 cells and dendritic cells. Each presenting cell will be infected by a virus. The virus then gets processed inside the cell and broken down by enzymes to produce peptides. Once processed, the virus is no longer able to harm the cell. The peptides then get displayed on both the MHC1 and MHC2 markers on the surface of the APC. The APC then travels up the lymph node to present the peptide. Differentiation and proliferation of CD8+ cells requires two signals. The initial signal comes from the binding to the APC, and the second comes from co-stimulatory molecules such as B7 binding to the CD28 receptor on the surface of the APC. The Th1 cell and the CD8+ cell initially bind to the antigen-presenting cell. Preceding the binding of Th1 to the APC, the Th1 cell gets stimulated by cytokines to produce a CD40 ligand. The interaction as well as the release of interferon gamma stimulates the production of a co-stimulatory molecule on the CD8+ cell, which binds to the CD80 receptor on the APC. The co-stimulatory molecule activates the CD8+ cell to become a cytotoxic effector T cell. The CTL exists in the lymph node and travels to the periphery into the site of infection. At the periphery, the cytotoxic T cell gets reactivated. It binds to the target cell via the interaction between its TCR and the MHC1 peptide on the surface of the target cell. Furthermore, the adhesion molecules LFA gamma and ICAM1 are used to create a stronger adhesion and a direct synapse for the effector molecules released by CTLs. Two molecules allow CTLs to perform their function: perforins and granzymes. Perforins allow granzymes to enter the cell by creating gaps in the plasma membrane of the infected cell. Granzyme B enters the cytosol through the ruptured membrane and activates caspases. The activated caspases activate second messengers and enzymes involved in the extrinsic apoptotic pathway. The cell undergoes apoptosis and the virus no longer has a host cell to grow or replicate in. The apoptotic bleb gets engulfed by APCs and destroyed by lysosomal enzymes. Once the cell undergoes apoptosis, the CTL moves on to another infected cell and repeats the process over and over again. In addition, CTLs release interferon gamma, which helps macrophages and T helper 1s to differentiate, express MHC1, and perform their actions better. It also interferes with viral replication, which further restricts the viral infection to move to other cells. As you can see, there are many interactions between all cells in order to mount an immune response. The immune system is a complex system and can be very specific to combat pathogens and mount an efficient immune response.